Thank you everyone for coming. I know it's Friday afternoon, 3 o'clock, so very nice to see quite a crowd here. I'd like to start by an acknowledgement to country. I begin today by acknowledging the traditional custodians of the land on which we are meeting on today, which are the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present. And I also extend that respect to Aboriginal and Torres Strait Islanders peoples here today. Okay, so it's my great pleasure to introduce Erandee Roberston, who has been our PhD student in the lab. This is her PhD completion seminar. So Erandee is a true COVID-19 PhD student. Erandee contacted me in November 2018 to investigate doing a PhD with me, so this is sort of a cold call. She has a BSc in Statistics and Operations Research, first class honours degree from Sri Lanka, which she got in 2016, and she worked as I had to look this up, Erandee, and I was like, oh, interesting. She worked as a market researcher in Sri Lanka for 2 years after that, and then contacted me to investigate doing a PhD. So she started with me in March 2019, so got in just before everything went pear-shaped. She was awarded a prestigious Melbourne Research Scholarship, so being an international research student, so you can imagine that Erandee is a well-performing scholar. Most of the time now when we take on PhD students, we take on PhD students who've been, at least in my case, RAs or you have a relationship with, but Erandee and I really didn't know each other at all. So we started from scratch, which is always interesting because you have to learn about each other, you know, and you're entering on a multi-year journey together. So that was interesting. So we had to learn to get to know each other. And then Erandee got married in 2020, and I have a lovely photo in my office from her wedding, which I really like. And Erandee's husband was in Sydney, and when we started having our first lockdown, Erandee left to go to Sydney, got very smart and just sort of ran and avoided the entire lockdown in Melbourne. And so Erandee was sort of having a good time with beautiful weather in Sydney whilst we were in lockdown. And so our first year, really the next sort of from the 6 months to the 18-month mark, I just saw Erandee online from Sydney. And I would just tell her tales of woe about homeschooling, etc. And, you know, it was quite interesting. And during that time, we developed the model that she'll explain to you today. So she came back to Melbourne in, I think, early 2021 and has been based here. And just a bit about her work. So I proposed to her, I mean, you know, Erandee's background is in statistics, a very strong quantitative person. And I proposed to her a crazy idea of mine that I hadn't been able to kind of convince anybody else in my lab to work on. And I thought it would be a good PhD project. So this is really all about searching for known rare inherited mutations using SNP chip data. And Erandee will explain what that means and how that works and why it works as part of her talk. And her undergraduate degree really made it a really good fit to try and attempt to do that. But Erandee's PhD, never mind that she achieved all of this, it's been a really interesting journey for me to observe, and I hope Erandee has a chance to reflect on this too. Like many other PhD students, she's picked up a huge amount of skills and assets along the way. So we noticed, for example, Erandee's R skills really improved along the way. She came to the lab with no background in genetics, so really had to get up to speed with that. Her writing skills have improved dramatically, and she'll admit that freely. And also her analysis skills, her ability to synthesize and do some very beautiful graphical presentations have really increased as well. And she's worked really hard to achieve those goals. She was second author on a recently published American Journal of Human Genetics paper, which is a really good effort. And we are going to write up her results that form the main part of this talk. But we decided, in the interest of time, given this is getting close to her 4th year mark, or we're in her 4th year, can't quite remember, that we better complete the PhD first and we'll write up the paper after. Before I hand over to Erandee, I just wanted to publicly thank Mark Bennett, who's been a co-supervisor, he was a postdoc in my lab, and he's been really very much supportive of the project and has helped Erandee with the really hands-on stuff, especially with regards to the program, which I couldn't really do anymore. So thank you, Mark, also. And Erandee, welcome to the stage, and we look forward to hearing the fruits of your labour. Thanks.
Thanks, Melanie, for a nice introduction. Thanks, everyone, for coming here in person and those who have joined online. It is such a pleasure to share my PhD research with you today, which is about developing an algorithm called FoundHaplo to detect individuals with rare disease variants. First in the talk, I'll take you through how my research idea was inspired in the first place, and the approach we took in developing the FoundHaplo algorithm, followed by a comprehensive simulation study I did to evaluate the performance of the developed algorithm, and then finally demonstrating how this FoundHaplo algorithm was applied and successfully detected individuals with disease variants. Many genetic diseases are inherited. Here I'm showing you few examples for Huntington's disease, FAME1, which is familial adult myoclonic epilepsy type 1, cystic fibrosis, which is a recessive disorder, and hemophilia. Huntington's and FAME1 are caused by short tandem repeat expansions, therefore known as repeat expansion disorders. Inherited genetic diseases like these have been there for centuries, affecting millions of individuals. Over the years, by analyzing multiple families like these, researchers often found out that affected individuals with the same disease variant have something in common. They share a genomic region around the shared disease-causing variant, which is also known as a haplotype. Now, haplotypes are going to be very important throughout the talk. They are the alleles inherited together from a single parent. Every individual has two haplotypes, one inherited from the mother, whereas the other inherited from the father. Now, instances where affected individuals share a co-haplotype are often associated with found defects, where the disease variant is believed to have originated from. Now, how do individuals who had inherited the same disease variant from a common founder also share a co-haplotype? I have illustrated that in a graphic here. At the top, you can see the disease-causing haplotype of a common founder and how that founder had passed down the disease variant, shown in red, along with the surrounding co-haplotype, shown here in blue. The size of the blue haplotype is different among individuals, and you can see it gradually decreases with time due to recombination. Recombination is the process by which DNA pieces are broken and recombined to produce new combination of alleles. Hypothetically, without recombination, all the subsequent generations would have inherited the same blue haplotype. But inevitably, due to recombination, individuals lose fragments of the original disease haplotype, as shown in this figure, which creates genetic diversity. Now, after many generations, when we take two distantly related individuals who had inherited the same disease variant from a common founder, we can witness a fragment of the original disease haplotype that is common to both individuals that had inherited from the common founder, along with the disease variant, without going through recombination. Now, preserved inherited genomic segments like these are known as IBD, or identity by descent segments, which are going to be the core of my research. The fact that affected individuals in present carry fragments of the original disease haplotypes show that we can predict individuals with disease variants by inferring if they have inherited known disease-causing haplotypes, because that means they would have also inherited the underlying disease variant as well. A bit of a logical thinking there. Now, this phenomenon of found defect that I have displayed in here is what inspired my research. These are some examples of found defects for ataxias, Huntington's, and types of epilepsies. For single and multiple found defects too, where the disease variant is believed to have originated more than once in history, presenting multiple unique disease haplotypes. There are hundreds of such found defects across inherited genetic diseases where our research idea can be applied. In fact, the two publications at the bottom are beautiful examples of found defects with contributions from our lab. I was a co-author in the publication at the bottom right, which got published in American Journal of Human Genetics, and was also chosen as a Nature Research highlight. You'll get to hear more of this study later in the talk. Now, since you have a reasonable understanding of found defects based on haplotype sharing among affected individuals, how are we going to use that concept to detect individuals with disease variants? First, I'll take you through how variants are identified in general. Variants can be detected using whole genome sequencing, where the entire genome is sequenced. There have also been development of bioinformatic tools that use WGS data to detect variants. For an example, these tools that I've listed here use whole genome sequencing data and help detect repeat expansion diseases. Variants can also be detected using whole exome sequencing, where only the coding regions are sequenced. As we all know, they miss out on non-coding variants. For an example, the FAME1 repeat expansion cannot be captured through WES because that's an intronic repeat expansion. WES is a couple of $100 per person, and whole genome sequencing is even more expensive. That's like $1,000 per person. On the other hand, the predominant data type in public databases would be SNP chip data, not whole genome or whole exome sequencing. Single nucleotide polymorphisms, or SNPs, are single base pair changes, and they are the most common type of variation. SNP chip genotyping takes a snapshot of the genome by genotyping specific locations with single nucleotide variants, as shown in this figure, thus making it cheaper than whole genome or whole exome sequencing, costing only around $50 per person. There are hundreds and thousands of individuals in public databases with SNP chip data in databases such as UK Biobank or dbGaP, which makes any tool that actually uses SNP chip data widely applicable. However, rare variants may not be captured on the SNP chip because, as I said, they simply capture variants at specific locations. In such instances, these missing variants can be imputed using genome-wide imputation tools, as shown in this figure, using tools like TopMed Imputation Server or Michigan Server, and there are plenty other genome-wide imputation tools as well. These tools use hidden Markov models to probabilistically predict missing variants with the help of a reference panel of haplotypes from public data sets such as HapMap or 1000 genomes. Unfortunately, imputation accuracy is known to be low when imputing rare variants. Since we know whole genome sequencing is anyway expensive, how can we capture variants that cannot be whole exome sequenced because they are not on coding regions? They are not on the SNP chip because they are rare. Difficult to be imputed accurately simply because they are rare again. Here we introduce our algorithm FoundHaplo that uses SNP chip data, which are commonly available, to identify individuals with rare disease variants with increased accuracy by leveraging on a database of known disease haplotypes. Here, one thing I would like to highlight early in the talk is that FoundHaplo algorithm does not try to detect disease variants genome-wide. Rather, it detects individuals with variants at a list of known disease loci. And the usage of SNP chip data makes this tool widely applicable, and we hope many individuals will be able to get the benefit out of it. The inference in the algorithm is done using a hidden Markov model, which I'll be explaining in the next few slides. Now, what are hidden Markov models? Hidden Markov models are used to predict sequence of our interest, which is hidden using another sequence that can be observed and related to the hidden sequence as shown in this figure. Hidden Markov models are widely used in detecting IBD segments, and not only limited to that, they have also been used in many other biomedical research as well. I'm simply showing you a few examples here. The hidden Markov model in the FoundHaplo algorithm tries to identify if an individual of our interest had inherited a known disease variant by accumulating evidence from the neighboring markers. To accomplish that, let's say this is a disease-causing haplotype that we have assembled, which has a known disease variant shown here in red. The haplotype is defined at a list of SNP markers. Say we have another individual, and this is the individual that we wish to test for this disease variant. Since we know disease haplotypes act as surrogates for known disease variants, the hidden Markov model in the FoundHaplo algorithm determines if the disease-causing haplotype and the haplotypes of the test individuals are identity by descent. For that, it takes the neighborhood around the disease variant locus, disease-causing variant locus, and compare the alleles and then update the IBD status at each genetic marker as it traverses around the disease variant locus. Therefore, there are two Markov chains in the algorithm. The waiting times in the hidden Markov model is taken to be the genetic map distance in centimorgans rather than the physical base pair distance, because centimorgan measures the probability of recombination between adjacent markers. Once the algorithm has traversed in the neighborhood, it could end up identifying that the alleles in the neighborhood is a poor match, or it could be a good match as shown in this example. If the alleles match, they could match by chance or as a result of identity by descent, and which is what we are interested in. Therefore, the FoundHaplo algorithm should be able to differentiate between the two scenarios. The hidden Markov model is developed in a hypothesis testing, where the null hypothesis is that haplotype around the disease variant is not identified by descent with the disease-causing haplotype. And therefore, under the null hypothesis, the hidden IBD status is denoted by a sequence of zeros. And alternate hypothesis implies that the haplotype around the disease variant is identity by descent, which is what we are interested in. Under the alternative hypothesis, the hidden IBD status is therefore denoted by a sequence of ones. The hypothesis testing is developed in a likelihood framework, and we calculate the likelihoods based on the alleles observed, conditional on each of the null and alternative hypotheses. The statistic in the algorithm is the FoundHaplo score, or the HEF score as shown here in the bottom, which is a measure of log likelihood ratio. The job of the HEF score in very simple terms is to differentiate cases from controls. So how are the predictions done in the algorithm? Let's say the HEF score of a disease test pair is high or precisely greater than or equals to a predetermined critical value. That implies that the neighborhood is identity by descent and have inherited from a common founder, satisfying our alternative hypothesis, implying that the respective test individual had inherited the respective disease-causing variant. I'll be discussing the derivation of the critical value later in the slides. Now that's the development of the hidden Markov model. Now before actually using the developed hidden Markov model, the original SNP chip data that we intend to use will have to be imputed to increase marker density, and also the phasing of the genotypes must be performed to create haplotypes, because inference in the algorithm is done based on inherited haplotype segments. In the next slide, I'll be explaining these two steps because they are going to be very important in making predictions. As I just said, original SNP chip data will be imputed using genome-wide imputation tools, which I described earlier in the talk, to increase marker density and to have millions of markers. The more markers the algorithm has, the better it can describe the haplotype, thus increasing the power. These genome-wide imputation tools could occasionally assign the incorrect allele to some of the markers. This is what's referred to as imputation errors. And our FoundHaplo algorithm should be able to accommodate such imputation errors. After imputing the genotype, the SNP chip data, phasing tools are used to probabilistically distinguish the maternal haplotype from the paternal haplotype and to create the two haplotypes of all the test individuals. And the imputation tools are generally designed for phasing as well and use a similar process. These genome-wide phasing tools could occasionally assign block of marker alleles to the incorrect haplotype. This is what's referred to as phasing errors, or commonly known as switch errors. Imputation errors generally occur around 1 to 2% of the times and could depend on how rare the imputed variants are, and also depend on many other factors as well. Phasing errors generally occur around 0.3% of the times, and similar to imputation errors, multiple factors could affect phasing errors as well. FoundHaplo has features inbuilt to accommodate these type of occasional imputation and phasing errors in our data. To cater to genotype and imputation errors, the algorithm has an additional layer that could accommodate a 1% of fixed error rate. Now, the test individuals are phased using genome-wide phasing tools, therefore prone to have these occasional phasing errors. To cater to phasing errors on the test individuals, as the algorithm traverses to the left and right, starting from the variant locus, whenever there's an allele mismatch on the haplotype that the Markov chain is in, it will switch to the other haplotype looking for a match. Now this switch helps the algorithm to cater to phasing errors on the test individual, and also it gives the best possibility for the test individual to share with the disease-causing haplotype. Now, number 3, phasing errors in creating disease haplotypes are avoided in the algorithm by creating disease haplotypes using affected individuals when we have more than one member affected in the same family. For an example, as shown in this figure, we can do trio phasing and create the disease haplotype shared by the affected offspring and by the affected parent without having to use genome-wide phasing tools that induce phasing errors. Now, this is a graphical representation of the FoundHaplo algorithm. I don't expect you to read anything on the slide. We have described, I described the steps of the FoundHaplo hidden Markov model in the previous slides. The algorithm takes mean allele frequency and the probability of recombination between adjacent markers and genotype imputation error of 1% as parameters in doing predictions. Now, up to this point, we have developed the features and the hidden Markov model in the algorithm. Now, before going forward, I'll briefly explain what the inputs and outputs of this algorithm are for your better understanding of how one can use this tool. FoundHaplo can use as few as one disease haplotype to do its predictions. In fact, it can use any number of disease haplotypes across multiple disease variants to detect individuals carrying those disease variants in a cohort. It'll output the individual HEFED score for each disease test pair across all the disease variants based on their IBD sharing, using which FoundHaplo will also output the combined HEFED score for every test sample across all the disease haplotypes tested for a single variant of interest. This combined score is believed to give more power in the algorithm as it leverages on multiple disease haplotypes. That means the more disease haplotypes we have, the better, especially when disease variants are identified with multiple found defects, because that means affected individuals in a cohort could have inherited multiple unique disease haplotypes. So we need more unique disease haplotypes in our setup to capture majority of those affected individuals. I'll be explaining the combined HEFED score value in detail later in the slides. Now, since we have developed the mathematical framework of the FoundHaplo algorithm, now novel algorithms like FoundHaplo require evaluation for performance before applying the algorithm on real data. Now, in the next few slides, I'll be explaining a simulation study I did to evaluate the performance of the algorithm. I used the European cohort of the 1,000 genomes haplotypes, which are publicly available and can be accessed from this link. I simulated the same haplotypes to act as disease haplotypes and test samples whenever necessary. Now, the very first step of the simulation study was to select a haplotype and consider it to be a disease haplotype by assuming it has a known disease variant. Now, that's our input 1. Input 2, a test cohort was simulated by designating fraction of samples as cases. For that, we select 5 haplotypes from 5 different individuals randomly and assume them to have inherited the same disease variant locus. Now, how are we going to do that? Since now we know that disease variants with such found defects are inherited along with the surrounding core haplotype, we replace the flanking haplotype around the disease variant locus of these cases with the already selected disease haplotype that has the disease variant, and rest of the cohort remains unchanged as controls. The size of this, sorry, I'm finding it difficult to find the cursor. The size of the simulated splicing segment is taken to be 0.5, 1, 2, and 5 in centimorgans on average to represent different distant relatedness to the most recent common ancestor. We simulated 33 known disease variants with known found defects. Any inherited disease variant with identified found defects can be used in the simulation setup. Simulations were performed to represent the 2 types of found defects that you know now, which are single and multiple found defects. And in every simulation, I simulated 1 to 10 disease haplotypes to compare in the simulated test cohorts. Now, after simulating our data, the next step was to add errors into the simulated data. I simulated genotype and imputation errors by swapping 1% of marker alleles to the other allele, and phasing errors were simulated by swapping a block of marker alleles to the other haplotype of all the test and control individuals. I did not simulate phasing errors on disease haplotypes because we assume they can be created using pedigree data without having to use genome-wide phasing tools that induce phasing errors. Now, after simulating disease haplotypes and test cohorts, which has simulated cases, which are the known truths, we applied the FoundHaplo algorithm to see if our algorithm could identify the simulated cases in each of these simulated test cohorts. Now, let's look at the insights of our simulation results. Here I'm showing you the results for FAME1 disease variant for an example, which represent both single and multiple found defect simulations. Now, before I mention that when we have multiple disease haplotypes, the FoundHaplo score represents the combined score. We investigated maximum, mean, and median of individual HEFET scores in the simulation setup, and based on the simulation results, maximum differentiated the cases from controls the best. Therefore, we are using the maximum of individual HEFET scores to represent our FoundHaplo score. The x-axis represents the distribution of the controls when only one disease haplotype is used in the calculation for now. Now, if our algorithm is capable of predicting cases, it should give higher HEFET score values to cases than controls. Here I'm showing you the results for the expected simulated segment of half a centimorgan for simulated cases. Here you can clearly see that the FoundHaplo algorithm works. It gives higher HEFET score values for the shortest IBD segment that we simulated. And the performance gets better as the size of the expected IBD segments gets larger, as we expected. Now let's see what happens if we increase the number of disease haplotypes used in the calculation. You can see the deviation between cases and controls now gets better as we increase the number of disease haplotypes. I used alternate shadings here so that you can easily refer to the 10 different partitions. I hope that helps. This deviation between cases and controls were not very clear when we had only one disease haplotype or a couple of disease haplotypes in the setup. Why? Because some of the simulated cases could have inherited multiple unique disease haplotypes, and as a result of that, they could share with other disease haplotypes that were not used in the FoundHaplo setup. Because of that, the algorithm loses those cases simply because it didn't have the right disease haplotypes or the unique multiple disease haplotypes in the calculation. That's why it's really important to accumulate as many unique disease haplotypes as possible in using FoundHaplo to get more accurate results from the algorithm. For higher number of disease haplotypes, another thing you would notice here that the FoundHaplo score of cases increases as the expected size of the simulated IBD segments gets larger. Again, I lost the cursor. Sorry, gets larger. So this deviation was not very clear when we had only one or two disease haplotypes or a couple of disease haplotypes. This shows that the performance of the FoundHaplo algorithm gets better when the simulated disease case pairs are more closely related to each other, when they have a more recent common ancestor, preserving larger IBD segments that can be better differentiated from the sharing of controls. Now, this behavior that I discussed with you is similar across all the 33 disease variants. I simply showed you FAME1 as an example to illustrate the findings of the simulation study. FoundHaplo score also vary by disease locus. Here again, I'm representing the score for both single and multiple found defect simulations and all the 1 to 10 number of disease haplotypes. Here also you can see the deviation between cases and controls. The same HEFET score here is represented across selected disease variants that we simulated. I'm not showing all the 33 variants here because that'll be a lot of plots. The point I'm trying to highlight here is that the FoundHaplo score vary by locus, as you can clearly see from the distribution. However, across all the 33 variants, even though I'm not showing them here, FoundHaplo algorithm gave higher HFS score values to cases than controls, allowing prediction of cases in a cohort. Now, since we demonstrated that FoundHaplo could differentiate cases from controls, how high of an HFS score a sample has to get for the algorithm to be able to predict as that sample share the disease haplotype and therefore carry the disease variant. Now, before going forward, I would like you all to look at the control distribution here, which is in black. You can see that we expect FoundHaplo score of the controls to be very low because we did not simulate them to share. However, you can see majority of the controls give low to moderate HFS score values, which is good. Some could share as high as some of the cases here. Therefore, when we derive the critical value, as I mentioned earlier in the talk, critical value is used to predict the respective samples to carry that, which is used to say that they carry the disease variant. Critical values are derived by using the FoundHaplo score distribution of a control cohort with similar genetic ancestry to the test cohort under investigation. The false positive rates in the predictions are minimized by taking the minimum critical value to be the 99th percentile of the 1,000 genomes, allowing an empirical false positive rate of only 1%. Here you can see even if I pick the same critical value of 99th percentile, the corresponding FoundHaplo score is not the same. It is not fixed. It could vary depending on the variant locus, or it could also vary depending on the set of genetic markers in the Markov chain. Because of that, every time a cohort is analyzed using FoundHaplo, a control cohort must be run to derive the critical value. And also the critical value can be increased from the 99th percentile to further reduce the number of false positives depending on how many samples someone is willing to screen, which I'll be explaining later in the application section. Now, this is the sensitivity of the FoundHaplo algorithm based on the critical values that we calculate. The sensitivity here represents both single and multiple found defects and also derived by using all the 10 disease haplotypes. I'm showing results here for 2 percentiles for comparison purposes. Here, the first highlight of the plot is that for the sensitivity of the algorithm increases when the critical value percentile is decreased to 99th percentile, however, could be at the cost of higher false positives. And also the sensitivity increases as we move along the x-axis. That means when simulated disease case pairs are more closely related to each other, preserving larger IBD segment, which is something we discussed earlier in the talk as well. This behavior is similar across all the 33 disease variants. On average, at the 99th percentile, FoundHaplo predicted 70% of cases sharing at least a region of 2 centimorgan and 80% of cases sharing 5 centimorgans or more. Here you can clearly see that the performance of the algorithm vary by disease locus. That means the performance could vary depending on how old the mutation is, which could impact the size of the IBD segments shared between affected individuals in present, and the performance could also vary depending on how unique the disease haplotypes are. With the simulation results and our setup, we demonstrated that the FoundHaplo algorithm works. It is capable of giving high HFS score values for cases than controls, allowing their prediction. The performance of the algorithm increases when cases are more closely related to the disease haplotypes sharing large IBD segments. And performance also increases when more disease haplotypes are available, especially when affected individuals have inherited multiple unique disease haplotypes. As I said before, the critical values can be changed depending on how many samples someone is willing to screen. Now, after getting satisfactory results from the simulations, we applied the FoundHaplo algorithm to detect individuals with a rare epilepsy variant. Here we are interested in a variant that causes genetic epilepsy with febrile seizures plus, also known as GEFS plus, which is an autosomal dominant epilepsy syndrome. We are interested in a point mutation in gene SCN1B causing GEFS+. This variant that we investigated has incomplete penetrance and relatively mild phenotype in most carriers. And also a found effect has been identified in European families for this variant. The main cohort in this application, I name it the GEFS+ cohort, is explained in detail in this publication in American Journal of Human Genetics. There are 9 unrelated Australian families with SNP chip data in this GEFS+ cohort, altogether 100 samples, and 32 individuals are confirmed to carry this GEFS+ variant in this cohort, confirmed using Sanger sequencing or whole exome sequencing. All the affected individuals in this cohort were identified to share a core haplotype of half a centimorgan around the disease variant, as shown in this figure, suggesting a common ancestor for all the affected individuals. And the common ancestor was calculated to have originated approximately 27 generations ago. This core GEFS+ disease haplotype was also found to be present in 1% of the European population anyway. Therefore, our FoundHaplo algorithm is likely to predict individuals that could carry the disease haplotypes, but not the disease variant. You'll get to see that when we come to the results section. Now, after preparing our input data, imputation and phasing was done using Michigan Imputation Server. And before applying the FoundHaplo algorithm, I created 5 disease haplotypes by pedigree phasing 5 offspring from the GEFS+ cohort. And these created 5 disease haplotypes were tested in the GEFS+ cohort itself, in the EPI25 cohort, and in the UK Biobank cohort. EPI25 is a part of EPI25 collaborative that has thousands of individuals with different forms of epilepsies, and majority of them are not yet solved for their epilepsy variants. Our third cohort, the UK Biobank cohort, is a large observational cohort of 500,000 individuals, and the cohort we analyzed here has around 200,000 samples with SNP chip and WES data. Therefore, this is a subset of the original UK Biobank cohort of 500,000 individuals. Using WES analysis, it was found out that there are 2 individuals with the GEFS+ variant in the EPI25 cohort and 74 individuals with the GEFS+ variant in the UK Biobank cohort. Now, since we already know the truths in each of these cohorts, what we did was a blind analysis to see if our FoundHaplo algorithm can detect the already identified individuals with the GEFS+ variant. Now let's look at the results of the GEFS+ cohort first. Here, this plot shows the 100 samples in the GEFS+ cohort, and samples are in descending order of the FoundHaplo score. The red dashed lines mark the 99th percentile of the 1,000 genomes European cohort taken as the controls. If we take the 99th percentile as the critical value, the predictions by the algorithm will be the samples that are above the 99th percentile, which is this subset of samples. Now, in doing predictions using FoundHaplo, we try to minimize false positives. Therefore, an additional step we can do is to look at the samples that are above 99th percentile, which is the minimum critical value we allow anyway, and see if some samples show even higher HFS score values and deviate from the rest of the cohort. So you can see that sort of happens somewhere around here, which I have marked in black dashed lines. Here we try to visualize outliers manually. With that approach, an alternative way would be to select this outlier point as the critical value because these samples look more promising than the rest. And we would also hope this outlier point will give less false positives than the 99th percentile. Now, the second plot shows the FoundHaplo score of the GEFS+ cohort in a solid violin plot and the controls in a dashed violin plot. I have only plotted the samples that are above the 99th percentile, which is kind of like the first level of filtering or base filtering that we are doing in the analysis. The HFS score distribution of a test cohort to a control cohort is a good indication of the strength of the signal of FoundHaplo. Here you can see the HFS score distribution of the GEFS+ cohort has shifted upwards compared to the control cohort because 32% of individuals in the GEFS+ cohort carry this variant. Now let's highlight the samples, the 32 samples with the GEFS+ variant to see where they lie in the distribution. They are marked in red here. Now you can see that FoundHaplo algorithm gave high HFS score values to majority of the cases, already identified 32 truths, which shows that our algorithm works and it's capable of identifying this variant in the GEFS cohort. If we pick the 99th percentile as the critical value, we'll end up identifying all the 32 individuals correctly to carry the variant, which is 100% sensitivity, with 4 false positives. If we pick the outlier point as the critical value, we are losing 4 true positives. However, it still gives a pretty good sensitivity of 88%. And you can also see that false positives have gone down from 4 to 1 as expected. Now, after getting satisfactory results of FoundHaplo algorithm in the GEFS cohort, now let's look at the results of EPI25 and the UK Biobank cohort. Straight away going into the plots because I explained how to interpret the results using the plots of the GEFS cohort. In the EPI25 cohort plot, I have again marked the 99th percentile and outlier point here. You may not be able to clearly differentiate the two because these 2 points are very close to each other. If we pick the 99th percentile here, again, FoundHaplo algorithm predicts both individuals correctly to carry the disease variant with 13 false positives. With the outlier point as the critical value, it still predicts both individuals, and now the false positives have again gone down from 13 to 7. Now, both these cohorts demonstrate the efficiency of our algorithm. Now, let's look at the last cohort, which is the UK Biobank cohort. The analysis of the UK Biobank cohort was slightly different to the other 2 cohorts because now this is a large cohort of 200,000 individuals. The results are plotted here in the same format as in other 2 cohorts. I have only plotted samples up to the 99th percentile because 200,000 samples is a lot of samples to plot. You can see the outlier point here, which is marked in black dashed lines, and all the 74 truths are shown here in red. Now, if we pick the 99th percentile again as the critical value, now here we are getting around 4,000 samples. That's a lot of samples and clearly an unrealistic amount to screen. FoundHaplo gives this many samples as predictions because UK Biobank is a large cohort of 200,000 individuals, so the algorithm will inevitably give many samples with high FFS score values. Now, with the outlier point as the critical value, it gives 120 predictions, which is a reasonable amount of predictions for someone to screen, and also a good sensitivity of 73%. If someone feels like even 120 samples is a lot of samples to screen, one can always pick the top n number of samples with the highest FFS score values. For example, if you pick the top 100 samples as shown here, you'll end up identifying 50 individuals in the UK Biobank carrying this rare epilepsy variant. I mentioned earlier in the talk that this GEFS disease haplotype is present in 1% of the European populations anyway. So you can see the false positive rates here 0.02 and 0.03 is much lower than that background sharing of 1%. So we are getting really small number of false positives from such a large cohort of 200,000 samples. Now, after going through the FoundHaplo results for these 3 cohorts, after getting satisfactory results, we compared our results with the genome-wide imputation tools. I tried to impute this variant using Michigan Imputation Server and the TopMed server both. Now, before going forward, I would like to say that genome-wide imputation tools are kind of like an alternative way to FoundHaplo because they also try to impute variant, not really disease variants per se, but variants genome-wide. The TopMed server is known to be one of the best imputation servers that has the largest reference panel of haplotypes and inevitably expected to give high accuracy than majority of other imputation tools. However, both these tools were not able to impute the GEFS variant in the GEFS cohort or in the EPI25 cohort at all. However, UK Biobank was able to impute the variant using Impute2 software tool, but the variant had very low accuracy, and as a result, it only predicted 1 individual to correctly carry the variant out of 74. Now, with FoundHaplo results, it predicted 88%, 100%, and 73% of individuals to correctly carry the GEFS variant in these cohorts. This shows that the locus-specific IBD approach in our algorithm has more potential than genome-wide imputation tools to detect rare variants in individuals, because FoundHaplo algorithm leverage on known disease haplotypes, whereas these genome-wide imputation tools gather data from general reference haplotypes, not really disease haplotypes. We also applied the algorithm to detect epilepsy variants such as FAME1, FAME2, and many other variants as well in the EPI25 and the UK Biobank cohort, and I'm still waiting for the confirmation of these results. They are yet to verify. These applications and the simulation study clearly demonstrated, I hope, that the FoundHaplo tool is an efficient tool in predicting disease variants in the individuals. However, it has its own limitations, and I hope I discuss some of them clearly with you. And there are some ideas that we have in mind that could further potentially increase the performance of the algorithm. For an example, a more sophisticated approach would be to model the genotype and imputation error rate as a function of minor allele frequency instead of taking a fixed rate across, because these errors are known to vary depending on how rare the variants are. For now, not accommodating phasing errors on the disease individuals kind of restricts the accumulation of disease haplotypes. So with the with modeling phasing errors on disease individuals, we'll be able to accumulate more disease haplotypes, and as a result, we'll be able to use FoundHaplo algorithms across many disease variants once adequate number of disease haplotypes are gathered. We have not modeled linkage disequilibrium in the FoundHaplo algorithm because we believe it does not have an impact on the predictions, because FoundHaplo tries to identify IBD segments larger than LD block size. However, with the modeling of linkage disequilibrium, FoundHaplo could increase the performance in identifying short IBD segments, much shorter than 1 centimorgan or half a centimorgans. In terms of what I'm planning to achieve during my PhD in the next couple of months, I have already coded up the algorithm in GitHub as our package, and I aim to make it public in near future. I've also designed a database to store disease haplotypes, and a MySQL schema is already developed. The aim is to make the MySQL schema public so that users can create their own instance of the database and they can manage their disease haplotypes efficiently and also run the FoundHaplo algorithm more efficiently. To our knowledge, there's no such database of disease haplotypes out there. And also to apply FoundHaplo algorithm across different diseases and many cohorts of interest. In summary, I briefly explained the FoundHaplo algorithm that I developed as a part of my PhD, which is an efficient tool in detecting individuals with disease-causing variants. And we discussed the mathematical framework of a hidden Markov model in FoundHaplo algorithm, and that it has also been implemented as an R package. Performance of the algorithm was evaluated using a comprehensive simulation study, and we applied the algorithm to detect disease-causing variants in cohorts of individuals. I briefly discussed the development of MySQL database to accumulate known disease haplotypes. We really believe that this database will help the accumulation of disease haplotypes, could be through collaboration at some point. In conclusion, FoundHaplo is an algorithm that can be widely applicable because of its usage of SNP chip data, and it has the potential to identify individuals likely to carry known disease variants who would otherwise be unlikely to undergo genetic sequencing or receive a genetic diagnosis. With that, I would like to conclude my talk, and I would like to thank my supervisors, Melanie, Mark, and David. Thank you so much for being, I can't believe the talk is finished. Thank you so much for being such amazing supervisors. You have guided me throughout, and Melanie, thanks so much for giving me the opportunity to come here and do my research. I still remember the day I received your confirmation email. That was one of the best days of my life. Thanks for looking out for my personal and professional development. You and Mark both made me a better researcher, I believe. And thanks everyone in the PhD committee, Matt, Paul, and Gordon. Your honest feedback was much appreciated throughout these 4 years. Those feedback and your encouragement kind of brought everything up to speed. Everyone in my lab, they all have been amazing mentors, guiding me throughout and sharing their scientific ideas. And my lab, I would say, made this journey a whole lot better. And I'm really proud to be a part of Barlow Lab. Everyone in all the Epi25 collaborators, especially Bron, who's the reason why we have the GEFS Plus application, which ended up in a really nice publication as well. Everyone in the division, academic administrators, and the scholarship I received from WEHI and from the University of Melbourne in support of this research, all the participants in UK Biobank and in Epi25, and all my family and friends, my parents, my siblings, my husband and my husband's family. They made these 4 years a whole lot easier for me. Without them and without all these people, I don't think I would have made it. Thank you so much, and questions are welcome.
Oops, yep, working. Excellent. Oh, yeah. All right. Thank you very much, Erandee. That was fantastic. It was just lovely to see it all together. So you did a great job. I just realised you have to design an icon for your art package, right?
Yes.
Maybe you can take some advice from Matt, who's an expert in this at some stage, and get some feedback. I have got a question on Teams, but any questions from the floor first?
Oh, thanks very much, Erandee, for that lovely presentation. Just to get going with a little clarification question, I must have missed your explanation of how you define the outliers when you were looking at the 99th percentile, and then you looked at outliers. Can you just tell me again what that definition was?
Yeah, the idea is because some controls could share with the disease haplotypes, we use the distribution of a control cohort to make predictions. So that's what the critical value is. So the minimum critical value I take from the simulations is 99th percentile. And I said in the talk, depending on how sample someone is willing to screen, let's say 10, depending on budget limitations, you can always pick the top n number of samples. Now the outliers, I came up with that as kind of like an additional step. If someone wants to further decrease the number of samples that they wish to test, it was done manually, not in a statistical method. It purely addressed the fact that if they can't screen all the samples that were predicted by the algorithm, it's some sort of, for an example, in the UK Biobank cohort, it gives at the 99th percentile a lot of samples. So you can, the next step would be to see if there are outliers that kind of deviate from the controls. And then if even that is too high, again, pick the top n number of samples.
Okay, but the outliers then are just your kind of visual assessment by looking at the distribution. Yes,
yes, true.
Because there are various ways to try and quantify
yes, and
that,
it would vary depending on the test
cohort. Way, of course. Yes,
no.
Yes. Okay, thank you.
Any other questions from the floor? No, I might just turn to, this is a question from Saskia. Have you any ability to go back to the GEFS Plus individuals and see whether any of the false positives with very strong FH scores have since experienced epilepsy?
If they have epilepsy?
Yeah, I think
it's the ones with the false positives. From what I remember, they do in the Epi25, of course, that's an epilepsy cohort.
They were recruited because of that.
Yes. Yeah. In the UK Biobank cohort, we, with that publication, I remember Bron checked the 74 individuals out of that, which individuals would have the epilepsy, but we really did not look at the false positives. We didn't do that yet. That would be interesting to do.
I guess the bottom line with the GEFS Plus cohort is we had the truth for the 200,000, and going forward, we're going to have the truth for the remainders as well. So the truth being, we can actually read off the variant status, which in a way is the right truth to match with the haplotype rather than the epilepsy status.
Yeah, that's true. Yeah.
Any other questions? Oh, Matt?
Yeah, thanks for a great talk, Erandee, and congrats on your R package and the database schema that you mentioned there sounds really useful. I wonder how that gets used in practice. So is the idea that a lot of these data sets are kind of behind closed doors, and so you really have to say, all right, we have to take this schema, and then just, yeah, if you can give me an idea about that as someone who doesn't work in this area directly. Thank you.
Yeah, for now, in terms of using the R package, the main, the very serious step would be to accumulate. I have put all the instructions on how to prepare the data sets, how to run it step by step. But for someone who's actually going to use the database, the main the serious concern would be to accumulate disease haplotypes, because for now they should be created using pedigree data. But once the model, somewhere in future, if the model can accommodate those phasing errors, any type of affected individuals can be used. I would say that preparation of some of the points would be really critical before actually applying the algorithm. And yeah, I hope that I answered your question. Yeah.
I think
Thanks.
I can act as an intermediary here. You're right, Matt. We've released a schema because of the privacy issue as well.
Yeah, that's
but
true.
You're going to populate a local one as well, which can be used, the ones that are public and are okay. But we figure there's going to be a number of applications.
Yeah. For now, the algorithm anyway can work without the database completely. You can just save the disease haplotypes in a folder and the algorithm can read from that folder. But the database would be a much methodical way of doing that. So the users have the option to create the database. If they are going to keep on using the algorithm, that would be beneficial for them.
All right. One more quick question. It's 2 minutes to, I know there's another PhD seminar. So you're the last one. Yeah. Thanks.
Hi, Erandee. Thank you for this really nice talk. So I was wondering if this algorithm can be implemented in complex disease variants, where not a single variant is responsible to cause the disease, but in fact multiple variants, and that has been distributed maybe outside your haplotype block. So can this be applied to that kind of situation?
Yeah, we haven't tested, but the thing is, the FoundHaplo algorithm detects not if an individual is affected or not. That's not what it's looking for. It's looking for the inheritance of the variant. If, let's say, disease A is caused by multiple genes, and if those genes have a founder effect, if, let's say, some of the genes have a founder effect or some of the variants have a founder effect, based on that founder effect, I would say that the algorithm can be used. It will only say that if that variant is inherited or not, nothing to do with other variants. I hope I answered it correctly. Yeah.
But for common variants you'd impute with, you'd be able to compute with normal methods. This is really in the space of
normally, yes, if they are common, yeah, you should be able to get good results from imputation tools.
All right, I think we have to wrap up there. So let's thank Erandee again for her completing seminar.
Thanks.